rods and cones contain something called rhodopsin.
Oh, rods and cones?
Yeah, they're different versions of rhodopsin. So when the light hits the rhodopsin in my receptors, that tells the receptors to start slowing down, turning off, hyperpolarize, whatever word you want. Okay?
It tells what?
Tells them to
shut up.
do this, right? Slow down. So what's going to happen to PDE when my rhodopsin is turning off my neurons?
Increase.
Let's see. If I turn these off, this goes on. So my PDE level should go up. I should be turning on my PDE by turning off my eye.
So the rhodopsin is actually decreasing the
Yes.
continuous demand?
Yes. So rhodopsin's going down, the PDE is going up. Right? Make sense? So it's the exact opposite of what you think. Make sense? You should be really bothered now. So I'm going to play with this a little bit.
So what's the...
Right? Let me show you one more little animation here to this concept. Went too far. Hold on. So this is trying to show you kind of in a static way. No, you never have to draw this. Here's my rods and cones, my photoreceptors, which contain that rhodopsin. And here, this is showing the frequency to my brain. So when you're looking at something, the brain's checking how often these cells are firing, the ganglion cells. And so if you change what it is you're looking at, the frequency to your brain changes. And the brain measures that as the lights came on, the lights went off. You flash, you're looking at books, whatever. So if you notice here, every once in a while there'll be a change in that frequency. It should be coming up soon. Right there. See it? The brain just said something. The brain interpreted that as, I just blinked, or you just opened the book, or something. So the brain is measuring the differences in the frequencies, not the actual frequency, but how it changed is what the brain's measuring. Makes sense? So again, the brain's interpreting, it's like your internet connection. How many hertz is it, is what your brain's interpreting. Make sense? So your brain's inventing a picture from a frequency of a neuron. So you should be really bothered by that. And then we're going to take you home to the really neurotic. You're going to go to therapy after this one. And that's this concept right here.
You said it doesn't change the frequency. It doesn't measure the frequency itself.
Right, it measures the difference.
Okay.
So the frequency is changing and tells the brain you did something different.
Okay.
Right? So if that frequency thing is what matters, which is what it does, this is showing a part of your retina, and we have different cones. You notice the cones here say plus, cones here say minus. So what happens in English is if the light hits these cones, the frequency might go up. If the light hits these cones, that might make the frequency go down through the same steps. Right? I'm changing the frequency. So let's suppose you put a bunch of light right through here. You'll notice that some of the cones will say speed up, some will say slow down. And that'll make a difference between the frequency you started with and the frequency you ended with, right? What happens if the light was just here? What would the frequency do? It'd go up because there are only pluses. If the light was only in this center, what would the frequency do? Go down. And the brain's going to measure those differences to figure out not only the light is on, but where is the light on my retina? Right? So let's do that math. So here's the frequency to my brain, down here. I'm going to turn on the light. What did the frequency just do? Try it again. You're watching. It came on. That tells the brain that these cones are getting the light, these ones didn't. Right? So those changes as the light is on my retina is what the brain's interpreting as not only that you're seeing, but where on my retina am I seeing it. Make sense? It's like a code. So then you're going to get this concept. As the light hits different parts of my retina, Those change frequency, and therefore you can tell where an edge is. When your eye looks at this picture, you don't actually see the picture. Your eye figures out where the edges are, because that's where you change the frequency. White's all the same, black's a different frequency, your brain says that must be an edge, because my frequency of my eye just changed there. Make sense? So in real life, you don't actually see pictures, you see edges in the picture. That should really freak you out. So you look at the picture of your kids, they're not there. The highlights of them are there. Because your brain doesn't know what's here, it only knows the difference here. Like this. So here's a black and white edge, like from textbook. Some of these ones will not be affected because nothing is changing. So this is not changing, this is not changing. But the ones in the middle, where the light is changing color, those ones change their frequencies, either up or down. The brain knows this is the edge of the image. Right here. This one's lighter, this one's darker. It must continue there because that's no different. Continues here, that's no different. It's like a big math problem. Yeah.
So the brain doesn't really care what the signal it's getting, it's
Right.
just when it changes that it wakes up
Exactly
and pays
right.
attention.
So to really
Other
figure
than that,
out,
it
it
doesn't
doesn't
really
care
care.
what the color is, it actually doesn't know what you're seeing. It just knows that it's a different color than before, and it's right there. Right? So, let's do this. That's why you, as I showed here, that's why you can read a textbook in bright light or dim light and the letters look the same. The brain doesn't know what the color actually of the page is. It just knows the H is a different color than the paper. That's all it knows. It doesn't care what the paper is. The H is darker than the paper. That's irrelevant. Right? So you get the difference. The contrast or the differences the brain sees. So that leads us to this ugly thing. This will I'll leave you with just to make yourself cry yourself to sleep. This is what you think you see. This is what the brain actually got for a picture. Because all the brain can detect are the outlines. And where do you see the yellow? Anyone see yellow? Right there. Why is that yellow? Because that's the center of your retina. You can't see color out here. So when you look at the taxi cab, that's what your brain got as the image. I get the outlines, I get the color in the center, and I get some kind of frequency change that tells me it's a yellow taxi cab on the street.
What?
So how do we interpret the color of the outer parts of the picture?
So what your brain would do, when your brain got this image, it'd say, I need a little more detail.
Right.
So to order your eyeball to change from looking here and tell your eye to scan around like that, recreate a new image each 1/60th of a second with the color. It remembers it and then invents a picture that you see based upon what remembers all those pixels as meaning in that 60th of a second. It's like your TV scanning down the image. That's what this is. But what your brain actually saw was corners and a little bit of color. And it invents the picture to fill in the detail. Paint by numbers is your brain. It just invents the color that should go there.
So I have a, oh, sorry.
So 60 frames per minute
Roughly.
is what cameras, slow-mo cameras, like to run at for
Yeah.
beautiful, pristine pictures.
Yeah.
That's why.
Basically, yeah.
That's kind of awesome. No, that is awesome. Yeah, that is really
Awesome.
awesome.
Yeah, cool. So just like a slow-mo camera or your TV set, right? Your TV's scanning, right? All those are individual pixels that turn on, but you see one image because that's faster than, the signal. Well, your brain doing the same thing with your eyeball. It's just forcing the eye to make rapid passes to fill in all the details. Right? Make sense?
So the optic nerve is the only nerve doing this?
Well, actually, the optic nerve is just a highway.
Okay.
Yeah, so the brain's interpreting the frequency of the nerve to interpret that.
And are the other nerves like moving the eye to help interpret?
Yes. So
Okay.
your brain then would then tell the other three that move the eye how to move to fill in the details it needs. Take home message, your brain sees, not the eye.
Okay.
Make sense? Okay. Make sense? So let me show you one more little thing on eyeballs. Just to really make you go drinking. Right?
Could have just bought it.
Which is this one here. Just a little brief. And we'll start ears. If you think about about your brain, those two things would show up exactly the same way in the optic nerve, right? As the outline of the line crosses your retina, the contrast would be seen. The brain would interpret the contrast and the color and realize the line's going this way. How would the brain know if the line is moving or you're moving? How does your eye know the difference? Because it's the same picture in relation to your retina.
Would it be equilibrium, your ears would tell
Partly
you?
your ears.
Partly proprioception, kind of giving it space and time.
So what the brain does, the brain realizes that it controls the direction your eyes move, right? So what the brain does is compare the picture it's getting with what it told the eye to do. So if the object is moving in a different relationship than the eye was told to move, it perceives it differently than if you reverse the two. So again, the brain not only is getting a picture, but trying to interpret what the picture means in relation to what you told your head to do. Freak out time now. So if it's doing this, then it knows the line is still. If it's, if it's doing something different, same thing with the size of the object. So the brain is using all the clues it has, your eyeball, your head position, the sizing, to figure out what it's seeing and what it's doing. Right? This is how optical illusions work, right? You play around with what the brain is trying to interpret. The brain misses it. Make sense? So you want to try this one?
No.
No?
I dare you.
Okay. Start at the center for a while.
It's moving.
Just start at the center. It won't do much. Right? So I'm going to hit stop. There's going to be a picture of a face that appears. That's not what we're going to pay attention to. You're going to pay attention to what the face picture does. Okay? Still looking?
It's gonna scare us all.
Gross. Weird.
Is it moving?
Yeah.
It's not moving on the screen.
Getting bigger.
Really? That got way bigger.
Yeah.
Yeah.
Then there was a starburst or something like that.
So that's one of the things you can do by trying to fool the brain into what it's perceiving. That's how those things work. Right? Same thing here. But, right? The brain can pick out that that is different than that just by just using some clues in your lifetime. So the brain learns as a baby that living things bend legs. Statues don't bend legs. Therefore, your brain knows that's a walking thing, not a walking thing. Right? So again, the idea is the brain uses its experience to figure out what those things mean on the same interpretation of the frequency. Go home and drink yourself silly, right? All right. So that gives us this picture here. So you don't have to draw this, but here's, what part of my brain is that for your quiz tonight? That's
Occipital.
my occipital lobe. And what do you do with occipital lobes?
Vision.
Vision. But if you read through that stuff, there's three things the brain's looking at. Where is the image? In other words, where is it in my eyeball? Is it up there in the center? Where is what I'm looking at? What is it I'm seeing? Is it a tiger? What is it? And then when did I see it? In other words, is the image moving or not? And your brain has to do that immediately when you open your eyes. Interpret that based upon biochemical differences in an optic nerve's frequency.
You know what's really interesting is babies, when they are around like 6 months old and they're not crawling, they don't get that
Right.
whole,
That's part of it, is you need
Feel.
experience to teach the brain how to interpret the differences.
yeah.
Would yellow exist if your mom didn't tell you there was yellow? Make sense?
Something would exist.
So take home message. Brain see.
Brain.
The brain is what you see. If I hit you in the back of your head and destroy your brain, you stop seeing. That's why. Okay. Let's show you one more thing here. To highlight just more things to make you all messed up. This is something in your lab manual tonight, but Saladin screws up teaching it, in my opinion. It makes it hard. But this is looking at your head. Here's your brain, right?
Mm-hmm.
Here are your eyes. Here's where you're seeing. You're seeing something up there. The colors represent which eye is looking at the image. So, my eye over here sees everything on this side and the center. The eye over here sees everything on this side and also the center. So you have binocular vision because both eyes can see the middle. Make sense? Your brain gets two pictures. If you look at the wiring, though, you'll notice the wiring is strange. Remember in lab, you have the optic chiasma, the optic cross. Look at the color of the wires and tell me what you see.
Some cross and some stay.
Some cross and some don't. So if you look carefully, the right side of this eyeball, and which is right here, and the right side of this eyeball both end up in the same part of my brain.
The right side of the brain.
The left side of this eyeball, the left side of this eyeball, end up on this side of my brain.
So your eyes are even.
So my eyes are split just like my brain, where there's a crossover for each eye. So the left half of each eye goes the same place. The right half of each eye goes the same place. Why would your brain want that?
So if there's damage, you could still see something.
Partly.
Homeostasis.
That's also with depth perception. Also, look at what's happening. The side of my eye here is receiving the input from this side, and same as on this one. So what it's doing is routing similar parts of your eyeball to the same place for interpreting. Excuse me. So I'm going to show you an animation of this briefly. It won't be that bad. Don't cry yet. You cry later. This is called the optic crossover, the optic chiasma. I'll show you the animation I want here. Here we go. Trying to find the right spot here. Gotta find the right spot. Oh, that's not it. All right, quick finger on that. There we go. Some reason, can't get that one. All right, let's try this again. Give me a second here. There it goes. Here we go. All right. All right, so let's just try to show in a weird detail. This is that thing where each eye is seeing sort of part of each other. All right? If you follow back, my eye here and my eye here cross each other back here. If you look carefully, where the optic nerve comes out is called your blind spot. You don't have any neurons there. If you notice, they're on opposite sides of each eye. So whatever this eye sees matches what that eye is missing. Right? What this eye doesn't see, this eye can fill in. That's one reason they kind of cross like that. Let's keep going. Here's the chiasma. That's where we cross over. So those wires then do the right side of each eye goes here, left eye goes here, so the brain is processing it as a unit. So each side of each vision is processed as one thing by your occipital lobe. Make sense? So when you read your lab manual about the optic chiasma, they simply mean that each eye crosses over half of its vision to the other side, not the full vision. So unlike the decussation, it isn't like right eye, left body. It's half of right eye, left body, half of right eye, left body, right body. Make sense?
Yes.
So that's just an anatomical feature we'll read about. But all it is meaning is that the eyes crisscross half their vision in the optic chiasma. And just so you as trivia, they line up differently depending where you're at, like that. So if you look at the wiring carefully, which I'm sure all of you are going to do all night, because you care, right? These two sides go here, they then branch the occipital lobe as a unit. So everything in my left field goes here, everything in my right field goes there. And therefore the brain's working as a unit instead of a variety of different neurons. That's the optic chiasma. Make sense? Mm-hmm. Anyway, so you have to draw it out, but all it is this crossover of each eye. Make sense? Questions on eyeballs?
Are we going to go through the rest of this?
No, we're not.
Okay.
Eyeball questions?
Nope.
Okay, what we're going to do for the next few minutes is do the chemoreceptors. So they're very quick and very easy to do. So we just do ear balls next week. Let's do some chemoreceptors. They're much easier to understand than this.
Good.
Good, I hear. You don't have to go home and crying with these ones. although you do. You may want to. So what do chemoreceptors detect again?
Chemicals.
Chemicals. And where in my body would that be?
Mouth.
That's my nose and my mouth or my tongue. And we have chemoreceptors. So what we're going to do is go through them relatively quickly because they're pretty simplistic. Let me show you some pictures of your chemoreceptors. You want the mouth first or the nose first? Okay, I heard mouth. All right. I don't care. You find your tongue. Where's your tongue? I lost my tongue. Here's my tongue. Okay. So here's your tongue. Here's my tongue. There's Mick Jagger. All right. So you had those bumps on your tongue, right? Long ago. Your mom told you those are taste buds. Your mom was wrong. Those are not the taste buds. Those bumps look like this. The taste buds are on the side of the bumps, not the top. So you get the lab, that's something you can remember. Here's a close-up of the side of the bumps on your tongue, which you would call the taste buds. If you look carefully at that picture right there, what do you see sticking out of that little onion-like thing? Hairs, gustatory hairs. Those are the chemoreceptors. So in your tongue, you have taste buds. Taste pores, you have a lot of words for them. And those contain the gustatory hairs, which in English means chemoreceptor. So you can say gustatory hair, you can say chemoreceptor, you can say taste pore hair. Sounds grotesque, right? But you have hair on your tongue. All right, so here's my hair. What do you think those hairs do? Why would you want hair sticking out of your tongue?
They grab everything as it goes by.
They grab everything as they go by. So they grab chemicals. So they grab, that's a good scientific word, chemicals in the saliva. Okay, explain that to me. Why did I have to put saliva there? Why couldn't they just grab chemicals willy-nilly?
Because it doesn't move around.
The enzymes break down? Or, no?
Transportation?
Was it?
This is like transportation, right?
Yeah, let's look where they are again. Where were they located?
Down in the water.
Down in the bottom of those bumps. How would the food on the top of my tongue get down in that little hole?
Saliva?
Saliva. So, you can't just taste up here. You have to get it down into those sides, which requires some saliva. You land on the hair. The hair then is connected to these cells, which is connected to this big wire. What is that wire there, that yellow thing? Sensory nerve fiber. So in English, that's a nerve. So they grab the chemicals in the saliva, and they send a message in the nerve. Where would I send this nerve message? To my brain. Does anyone remember reading or hearing which part of your brain does taste?
Gustatory.
Gustatory center, which is located in what lobe?
Temporal.
Usually the temporal lobe. It varies a little bit depending on what is it pain taste or good taste. But you swallow a Snickers bar or whatever, the Snickers chemicals wash over your tongue because it's so good, land on the hair sends a message to my brain and say, that is a Snickers bar. How does your brain know it's a Snickers bar?
It's a
So it told
memory.
So I told you what, same thing. So let's go through the tastes. There's only about 5 chemical types that you can grab. You learned these way back in middle school. What were the 5 tastes that they taught you?
Sweet,
So you have
Salty,
sweet,
salty,
savory,
salt,
savory.
sour,
Bitter?
bitter. Sometimes sour is called acidic. Bitter is called bad. And then there's a savory,
And then
which is
the
also called
meat.
richness,
And then that meat one.
which is kind of that. And there's a metallic taste, licking a nail. Right? Not Metallica, that's different. Right. So, you swallow a food, in theory, you have 1, 2, 3, 4, 5, 6, depending on who you talk to, different chemicals you can grab out of the food. So how do you know it's a Snickers bar and not roast beef?
difference.
Because you have sweet.
The different combinations of these 6 things. So again, the brain is interpreting a combination of signals to say that's roast beef, that's a Snickers bar. Right? So again, the brain tastes, the tongue just sends the message. The brain has to interpret. That's why I may think some food's awful tasting, and you think some food's great tasting. It depends on your brain's interpretation of these combinations of flavors. Right? Make sense? Although most everyone hates bitter. Right? But, so your brain is interpreting basically 6 things at some combination. Yes?
So are these 6 things attached, are they connected with their own receptor and only those receptors are
In theory, yes.
lighting when, I mean, that's how the brain knows.
Right.
It's like, oh, the sweet receptor,
Yes.
talk to me.
There used to be that brain, that tongue map. Remember when they had the tongue and they drew? That's all been false. That was wrong. It was never true. They don't just repeat it. What they found out was, yeah, different parts of your tongue are coded for different flavors. The brain just knows number 5 means sweet. Number 10 means this. Number 3 fired twice. That's sour.
Okay.
Right? And there's also a different receptor, which is pain. That's not a chemoreceptor. Those are separate receptors. So, the boiling hot Thai food that melts your mouth, it isn't one of these. It's that one, which the brain interprets as spicy, basically. But your brain takes this and says, that's Thanksgiving, the combination thereof. Make sense? It's a very simplistic system. You're just coding different flavors in different ways. And you have to teach the brain what those mean in terms of Snickers bar. Right? So what do you think your nose would look like? We did a nose.
Hairy.
Maybe a hairy nose. Let me show you your nose. Okay, we're not talking about the hairs in your nose that you pull out. We're talking about the hairs way up there with your nose. Way up there. Right here. Okay, it's exactly the same biology as your tongue. You think your tongue in your head. Right? We have hairs, which are chemoreceptors. These times called olfactory. Each one grabs a different chemical. So when you inhale a skunk, it lands on different hairs. Those go to the brain. The brain interprets that as, that stinks. My BO would hit on different hairs and go to your brain and says, he stinks. Chanel number 5 would land on different hairs, and your brain interprets that combination as, that stinks. Right? Make sense? It's the same exact mechanism of using hairs to grab chemicals out of the air and send a message to your brain. Well, let's go through it. How many different types of olfactory hairs do you have? We know we have about 6 or 7 tongue hairs. How many nose hairs do you have? Lots. Lots. So our olfactory hairs, equal infinite, we believe. We don't know because we don't know what you don't spell, right? But we believe there's a lot more hairs for smells than there are for tastes. Okay? So you can prove that. Try not smelling something. Right? Pretty much everything you can think of, you can smell and discern. You can discern one perfume from the other perfume, right? Mm-hmm. Right? But it's hard discerning a Snickers bar from a Milky Way. Right? They're roughly the same. So this one is much more acute. You can smell better than you can taste, more accurately. Yes?
Can you not smell anymore? I mean, theoretically, it sounds like you can't smell if you don't have any hairs.
Yes?
Is that true?
Burn them off.
Yes. So if, for example, you inhale ammonia, because you work at the hospital, ammonia kills off the olfactory hairs if you breathe it deeply. As you lose these hairs, you lose the acuteness of the smell. So you lose your ability to smell certain things as well as you could. Same thing with the tongue. Let's do the flaming hot pizza concept, right? You go have hot pizza, and one day the hot cheese goes under your tongue and begins to burn off pieces of your flesh. Am I the only one that admits to doing that? And then what do you think happens to little gustatory hairs when lava pizza hits them?
They
Burn
burn
them, and what
off.
happens to your taste?
It goes away.
Go away. But since you have a whole lot of those hairs and a whole lot of tongue, you don't notice it. You do that over your ages, Or as you get older and neurons die,
Yeah.
grandpa's complaining about nothing tastes right anymore. That's why he's pouring the salt out of the beaker, right? Because you're losing the ability to discern what you taste and smell.
So when you burn your tongue and the
Yes.
little hairs get burnt, they don't rejuvenate?
Only, well, if they're dead, they don't. If they're just damaged, they can.
So
It's
like,
also one reason they're down under the surface, right? You gotta get hot flaming oil down here, not just on the top.
so is it the same concept like if you burn your tongue at the beginning of the meal, you really can't taste the rest of the meal
Yeah,
very
it's kind
well?
of a pain thing. Yeah, it swells up and plugs those.
Oh, okay.
Are these
And
two
this-
related, the mouth and the nose?
Yes. In fact, your brain views them as the same thing, because where do you go to smell? What part of your brain does olfaction? Take a guess. Usually your temporal lobe. It varies, but it does a detour. Before it gets to the temporal lobe from either one, there's a little place you visit first, which is your limbic system. You go to your limbic system and then to your temporal lobe. What was your limbic system for?
Emotion.
Memories and
Emotion.
emotions.
So, perfume industry lives on this. You smell Chanel number 5, you get a feeling when you're smelling Chanel number 5. Yes.
I read an article last term that said people who lose their sense of smell sometimes get depressed at higher rates than if you lost another sense because it's tied in your emotions and your memories.
Exactly.
And so it's like you start to feel sad, but you don't really know why. It's because you're not experiencing,
happiness.
yeah,
Yeah.
sad.
Yes.
So with the two of them tied together,
Uh-huh.
I'm assuming that's why, like, if you have a cold or you're stuffed up, you can't taste as well, but
Very good.
what exactly is going on?
So if you have a cold, you plug off the nose. The brain loses that information, so all it has are the 6 tastes, which are less acute. Because what the brain does when you smell food is it tastes the food and adds the smell on top of it to interpret what you're eating.
So it
You
it
lose the
really
smell,
relies
all you
on
got
those
taste, which is why every food's bland when you have a cold. Every rodent in the restaurant industry knows this trick, right? The food has to smell good for it to taste good. And they're not directly the same scent, your brain interprets them the same way. Ever eat next to a sewage plant?
Gross.
You won't, right? It could be great food, you're not gonna go there, right? Because the smell will overwhelm what you're tasting. Same concept. So the brain views chemoreception as the same, no matter where it's from, basically.
So does smoking kill the receptors then?
If you're a smoker, not any of you are, right? Let's make you a smoker, right? So smoking, as you well know, kills off things, including the olfactory hairs. If you smoke a lot, what does food taste like? Tastes crummy, right?
Tastes like the
Because
salt
you can't
shaker
smell
that
the
you
food
shake.
anymore. You also don't smell your own stink. That's more probably habituation, also because you don't smell it anyway. Also in perfume, that's why grandma keeps pouring the perfume on, right? The whole bottle on her. Makes sense? So, chemoreception, it's very simple. Just go to your temporal lobe and interpret what neuron is saying what. That's the knowledge. Versus the eyeball, which is all make-believe. Makes sense?
So that's why the first bite of Hot Pockets tastes really bad, and then after you burn your tongue, you can bear the rest of it. In fact, you want to.
Tell me the other way. I think the first one's bad, I think it's better, but that's good.
Yeah, there you go.
Or better than worse. Yep, exactly. Yes.
As the kind of suggestiveness between people, oh, this is spicy, oh, it's not really spicy. Is that a difference in the
Yes. In fact, there's different genetics in how many taste buds are on your tongue. And there's a genetic difference between what receptor numbers there are on the tongue. So your brain gets different levels of signal, and there's a difference in how your brain is taught to agree with the signal. So the person who can get nuclear Thai food and be fine, a person like me who looks at it and gets sad, there's a genetic difference in how your brain interprets all this. They have super tasters and low tasters based on the number of hairs.
So someone that likes like really insanely spicy food is, and someone who doesn't like it as much, or a little bit of spice is a lot. The person that can handle all that spice just have less spice receptors, so it's really the same, but someone likes it more because they don't have as many spice receptors, so it's not killing them.
That's right. So the difference is how much pain am I tolerating? And then there's the difference of how much pain does my brain want to interpret. And those two things combined are the signal I get and the interpretation combined to make me melt when I see salsa.
So then that development in evolution, would that come from, the people that develop less receptors to the spicy food actually helps their existence somewhere, because they can, more are the ones that have more receptors, so they don't like to eat it as much, it's like,
Hard to say.
chemical makeup.
I'm trying to think if none of us were there, but the argument is, you adapt to certain cuisines based upon what kept you alive. And then, yeah, so people who couldn't taste the bad poisonous tansy ragwort weed all died. The ones who could taste the bitter tansy stayed alive, so all of us can taste alkaloids really strongly. Dry aspirin, ever try to swallow a dry aspirin? Right, you want to retch, because that taste is entirely bitter, it'll kill you. So your brain immediately says, if I get bitter, I'm going to make you gag. Because over evolutionary time, bitter things kill you, like alkaloids. Things that were sweet, people crave because sweets burn through lots of calories, right? So everyone likes sweets. Right? So yeah, so there is an evolutionary kind of pattern to it. But why some people like salsa and some don't, I don't know, did salsa kill people once? But you get that, you know, some pasty white European may not be able to handle the Latin food because over time your parents never had salsa. But it's hard to say where evolutionary time is salsa was kind of bad.
So you may have answered this while it was out, but, so like after I had my daughter, I was able to tolerate spicy food more than I was before. Like I couldn't even have onions before, and now I'm like,
Yes, pregnancy changes the number of receptors and the interpretation. That's why you smell so much when you're during pregnancy. It's because your brain amplifies the olfactory hair in their interpretation. So you can smell the baby.
But so then you have a leftover.
Yes, and then you gain the advantage of smelling better after baby.
So it doesn't change the number of them, it just changes the response from them?
Yes,
Okay.
more acute. Make sense? So, see you in lab in a while. Thank you for being here.
Is it going to be a while, or is it going to be in 10 minutes?
20 minutes.
About 20 minutes, I think.
Okay.
It's all set up, so I can start it.
Oh, it is.
Stop you and call and cry and let your brain interpret whatever you think is I love new classes. You don't know. You're going to sit there and bite your nails. Remember, I don't exist, right? This is an illusion.
So my grade